I also want to thank the organizers for putting together such a fantastic conference. It's really great. I'm excited to be here and share some of our work. So yesterday we heard some great talks about membranes and condensates from the theory side of the bridge. And today I'm going to share some of our works from the experimental side of how we're trying to understand the ways in which membranes and condensates interact with and influence each other. So before I start, I just want to acknowledge the people in my lab who did the work that I'll be sharing. So most of this work was done by a graduate student, Nick Lea, who's actually the first person to join my lab at MIT, and also two postdocs, Xiaoheng and Maria. And a few pieces of what I'll be presenting were done in collaboration with Petra Schul's lab at the Max Planck. So I'm an experimental biologist, and in my lab we use a combination of biochemical reconstitutions and cellular experiments to study condensates, and we're particularly interested in the role that condensates may play in regulating signal transduction in mammalian cells. So with biochemical reconstitution, these are very minimal, simple systems. We're purifying proteins from E. coli or insect cells. We're putting them in test tubes and we're observing what happens. Often our reconstitutions are somewhat complex, so usually we're looking at maybe 5 to 10 proteins, different types of proteins in combination, either in solution or often in the context of a lipid bilayer. The benefits, strengths of reconstitution, in my opinion, are they're really powerful for determining if a specific set of molecules are sufficient for a process of interest. They're very powerful for dissecting mechanism because we have complete control over the experimental parameters, unlike a really complicated cell. And they're really great hypothesis generating approaches. So they can generate hypotheses that we can then go and test in cells. With cells, of course, cells are way more complicated than a test tube. So the questions are always like, are these things we observe in this simple system really what's happening in the complex cellular environment? In cells, we can determine which molecules are necessary for a specific process, which is much harder to do kind of in a test tube. We can test these hypotheses and models that we've developed from simple systems and then study our molecules in specific cellular context or disease context, because depending on the cellular context, the behavior of these molecules might change. And really in our lab, we kind of iterate between these approaches, and it's this ability to go back and forth that we think allows us to deeply understand condensates in this case. So today I'm going to talk about membranes, and we heard a great introduction into membranes yesterday. And I really think, well, we just heard perhaps condensates are also fundamental to life, but really membranes are fundamental to life. All organisms have membranes, and perhaps all organisms also have condensates. This is all very exciting. So at the very least, every organism has a plasma membrane. This is my favorite membrane, the eukaryotic plasma membrane. This is a model from a simulation, computational simulation from a group at Oxford. And what you can see here is that the membrane is a complex environment. There's a lipid bilayer with lots of different lipid species that have distinct chemical properties. There's many different types of proteins embedded in the bilayer, receptors and channels. There's also proteins on the cytosolic side of the membrane that are transiently associating with the membrane, including an actin cortex. So the cytoskeletal filaments form this like dense mesh that sits next to the plasma membrane. And so membranes have a lot of really interesting properties, which we already heard about throughout this conference. They are a two-dimensional surface, and we've heard how really interesting physics and chemistry can happen on surfaces and at interfaces. So membranes are a very large surface in the cell. They contain this complex mixture of macromolecules, including proteins and lipids. Often the molecules in the membrane are rapidly diffusing in 2 dimensions. And something I won't talk about, but I do think is interesting, is they have a really wide range of topologies, like negative and positive curvature, and the topology can vary a lot. The experiments I do look at sort of flat membranes, but I think moving forward, it's really interesting to think how curvature can also influence organization. For those of you who don't think about the plasma membrane, I would also implore you to realize that membranes are everywhere in eukaryotic cells. So this is a multiplex movie time-lapse of a human cell with 6 different membrane compartments labeled from Jennifer Lippincott-Schwartz's lab. And what you can see is just the cytoplasm is full of different membrane-bound compartments. Particularly the ER is huge and dynamic. So I think in the condensate field, we have a tendency of thinking of condensates and membranes as these distinct compartments, so different ways of organizing the cytoplasm. But in reality, these compartments are in the same space, and they're often probably interacting with and influencing each other, as we've already began to hear yesterday. So in my research, I'm really interested in the plasma membrane, and particularly how receptors that are important for regulating signal transduction are spatially organized on the plasma membrane. So we've known for a long time in mammalian cells that signaling, so this ability of a cell to sense information in the environment and then transduce that information into the cytoplasm, this is often regulated by receptors, and often this requires these receptor molecules to undergo a change in their organization. So receptors will often cluster with hundreds or thousands of copies of similar receptors, and also recruit a specific subset of cytosolic adapter proteins and signaling molecules to form these signaling clusters that can be hundreds of nanometers or even microns in scale. And we know for many signaling pathways that a robust activation of signaling, being some change in internal biochemical activity of the cell, requires this cluster formation. And so I've, for a long time, been interested in understanding how this spatial organization arises and how changing the spatial organization can actually change the biochemistry in these signaling pathways to influence cellular function. And what's been very exciting over the last decade is we've begun to appreciate that phase separation can contribute to this type of organization at the plasma membrane. So there are many different types of receptors and diverse cell types that can be organized through phase separation that is often driven by interactions between the cytosolic domain of the receptor and cytosolic adapter proteins. So this is often phase separation driven by protein interactions. And so we heard, so I'm not going to go through all of these, but we heard yesterday a little bit about Z01 and Claudins at tight junctions. What I'm going to tell you about today is mostly work that we're doing looking at the integrin receptor, which is a receptor that binds to the extracellular matrix. So this is proteins secreted in the environment like fibronectin and collagen. So this type of adhesion is really important for animal cell tissue formation. And I'll also talk about another receptor, a cell-cell adhesion receptor called nephrin, which I worked on earlier in my career. And this is really important in your kidney, actually how your kidney functions. So it allows cells to form these really tight cell-cell adhesions that actually have this function of filtering blood. And so what's interesting is by sort of comparing different biological systems, some of the features are similar, but also sometimes they're different, and you can learn a lot by like understanding how are they similar and how are they different. And so that's what I hope to tell you today is a few different observations in these different systems that we've been making, and how this is all sort of synthesizing to inform how we think about the role of membranes in this protein-driven phase separation. Okay, so I'm going to sort of talk in the scaffold and client framework. So we've sort of heard this also throughout the meeting that when thinking about condensates, there are molecules that are scaffolds for the condensate. These are the molecules that are actually undergoing phase separation to drive the formation of this compartment. So a scaffold, in my definition, is required for phase separation. It's often highly concentrated within the condensate, but then there's also a lot of other proteins that come along for the ride. So the clients can be recruited to the condensate by directly or indirectly interacting with the scaffold. They can often be very important for the function of the condensate, but they themselves do not undergo phase separation. And they can often be maybe less concentrated than the scaffolds. And so this is also a complex, like an imperfect framework, and we just heard like sometimes it depends on the context. A molecule can be a scaffold in one instance and a client in others, but it's just like a way to try to think about these different systems in a more qualitative way. So the first observation we had when I started studying integrins was that receptors can, of course, be both scaffolds and clients. And so the nephrin is a receptor that I would classify as a scaffold. Its interactions are actually required for the phase separation that we observe. Nephrin contains 3 binding sites for an SH2 domain on an adapter protein that also has additional binding sites for another adapter protein. So this is a highly multivalent system. Each protein has multiple binding sites for the other, and you actually need all 3 proteins to interact with each other to observe phase separation at sort of cellular, like physiologically relevant concentrations. In contrast, integrin is what I would classify as a client receptor. So the integrin receptor, the cytosolic domain of this receptor, only has a single binding site for adapter proteins that are in the cytoplasm. And it's actually the cytosolic adapter proteins that are driving phase separation, and we've identified multiple scaffolds in this system that are somewhat redundant with each other. The one I'm going to talk about the most is this protein FAC, which is a kinase, focal adhesion kinase. So it's the cytosolic adapter proteins that actually drive phase separation, but they can interact with the receptor. Okay, and so what does this look like experimentally? So we can look at our biochemical reconstitutions. We make a supported lipid bilayer. In this experiment, we have a very simple lipid composition. So it's just a bilayer containing phosphatidylcholine. And then we include some nickel chelating lipids that allow us to attach his tag receptor proteins to the bilayer. And then we can also add our cytosolic adapter proteins to solution and observe what happens. So when we have nephrin on the bilayer and we add these two adapter proteins to solution, we observe the formation of these membrane-associated condensates. Similarly, if we do the same experiment with integrin as a receptor, we attach integrin to a bilayer and we add this sort of cytosolic adapter proteins. We also observe the formation of these membrane-associated condensates. In this image, nephrin is fluorescently labeled, and in this image, integrin is fluorescently labeled. So they're both forming these membrane-associated condensates. You can see the nephrin's more concentrated or enriched in the condensate, whereas the integrin is much less enriched in the condensate. And we think that's probably because the integrin is acting like a client. But this made me question, well, what is the role of a receptor if it's not actually driving the phase separation? Can it actually, control or regulate condensate nucleation if it's not itself have a propensity to phase separate? And another observation, I guess, to show you is if we remove the receptor. So in the case of nephrin, again, it's a scaffold. It's required for phase separation. So if you just take the cytosolic proteins in solution at micromolar concentrations, there's no condensates forming. So you really need the receptor in the system to observe the phase separation. In contrast, if you remove the integrin receptor and you just have the adapter proteins, you still form condensates at micromolar concentrations. And so this led to the question of why do you even need the receptor? What role does the receptor play in regulating this? And so to experimentally test this, we can make a bilayer that doesn't contain any receptor and compare that to a bilayer that has the receptor present. And we add the cytosolic proteins and we ask what happens. Do we see condensate formation in both of these cases, or does having this client receptor present actually help favor phase separation? And we kind of heard about this yesterday, so this perhaps is not a surprise. This is very similar to Z01 and Cloudens and how what was described yesterday. But we see that when we use really low concentrations of these proteins, so this is actually the physiological range of these proteins in the cytoplasm, we see that we actually don't see really condensate formation at all without the receptor. It's only when the receptor is on the bilayer that we start to see the nucleation of these condensates occurring. And we can quantify this over time. So on an empty bilayer, over the course of 10 minutes, there's very little sort of condensates nucleating, whereas if we have the receptor on the bilayer, we start to see condensate formation pretty quickly. If we increase the concentration of the sort of cytosolic adapter proteins to concentrations where they do spontaneously phase separate in solution, we still see the effect of the bilayer. So we still start to see, without the receptor, some condensates on the bilayer, but when you have the receptor present, there's much more condensate nucleation. And if we quantify this over time, it's really accelerating, having the receptor on the bilayer is accelerating the nucleation of these condensates. So client receptors can promote condensate nucleation on membranes. And we heard really nice theory yesterday kind of explaining how this sort of membrane surface might be favoring this. Another interesting observation with these proteins that makes the system more complicated and interesting is that a lot of these cytosolic adapter proteins bind directly to the lipids themselves as well, in addition to binding to the receptor. So for example, FAC, this protein that I've talked about, binds directly to a lipid species called PIP2 phosphoinositide. So this is a lipid that has this inositol head group, and it has these residues that get phosphorylated and dephosphorylated by kinases and phosphatases. So the lipid composition itself is being actively regulated by the activity of kinases and phosphatases, and when PIP2 is present, many of these adapter proteins can bind specifically to this PIP2 lipid. And so we wondered what would happen. Can the lipid composition maybe also play a similar role as a receptor to sort of recruit phase-separating proteins to the plasma membrane and potentially regulate condensate nucleation? And so to test this, we took a bilayer that just contained phosphatidylcholine, no receptor, so just lipids. and compare it to a bilayer that contains 2% PIP2, which is in sort of the physiological range of the concentration of this lipid on the plasma membrane. And here we're just looking at this protein FAC, which does phase separate on its own. And so if we have a 1 micromolar concentration of FAC, we can clearly see that although we see condensates forming on the sort of bilayer that doesn't contain the lipid that it binds to, we see many more condensates in the bilayer containing PIP2. And so this is quantified here. So again, having a lipid species that a protein can bind to on the bilayer can also favor condensate or sort of accelerate condensate nucleation on the bilayer. Another interesting example of how lipids can regulate condensate formation in a slightly different way we observed with this protein talin, which is another adapter protein that can bind to the integrin receptor and promote receptor clustering in cells. We find that talin can also phase separate. So another theme is many of the adapter proteins in this pathway all phase separate and seem to have this shared characteristic. And talin is a highly structured protein. So this is actually an example of a protein that doesn't really have disorder. It's mostly alpha helical, but we see when we purify it that it can form condensates. And what we found is that it's really when it's in an open. So talin is maintained in a closed conformation in the cytoplasm, and then it has to undergo a conformational change where it kind of unfolds and opens up to reveal sort of the binding sites for different proteins. And it's when talin is in this open conformation, that's the conformation that can undergo phase separation. So when we purify talin and we favor the open conformation through a variety of ways, we see that we can form these condensates in solution. So here we have 3 micromolar talin, which is pretty high, like non-physiological concentrations, and 20% methylcellulose as a crowding agent to sort of force this protein to form condensates. But the interesting thing is one of the things that favors the open conformation is binding of talin to the lipid. So PIP2 binds to this firm domain, and that helps to stabilize the open conformation. So the presence of this lipid can shift the conformational equilibrium towards the open conformation. And what we see in experiments, if we add talin to a bilayer containing PIP2, is that now we can see condensates nucleating at lower concentrations and without any crowding agent. And what we see on the bilayer is that we have talin nucleating or forming a condensate, and we also can see that the lipid itself is being concentrated in this condensate as well. And so here the lipid is not just concentrating the protein at the membrane or recruiting the protein to the membrane, but it's also changing the conformation of the protein to a conformation that favors phase separation. Okay, so all of this together in the case of integrin regulation is contributing to this sort of working model that we have where we think all of these proteins are expressed at pretty low levels in the cytoplasm, so sort of like nanomolar concentrations, and that at least in our experimental biochemical experiments, this is below the concentration required to see phase separation in a test tube. And so there's no spontaneous phase separation in the cytoplasm. It's only when you have the receptor in a conformation that can bind these proteins and or lipids in the membrane that the proteins can recognize, where you start to recruit these proteins to the membrane to then leading to phase separation. So we really think that it seems like the receptor and the membrane are controlling when and where this phase separation occurs. So this is leading to sort of the assembly of this receptor into a higher order cluster that is then, we think, important for downstream signaling. So next we wanted to test if these condensates might also regulate signaling, and we've been looking at this kinase, focal adhesion kinase, or FAC, which is one of the components of this condensate. So one of the functions of these integrin condensates is to activate kinases. So you can see here, this is an antibody that recognizes phosphorylated tyrosines, and that these condensates are enriched with these phosphorylated tyrosines. And this is in part due to the activity of this kinase FAC. So FAC is a kinase, a cytosolic tyrosine kinase. That means it's an enzyme that, phosphorylates other proteins. And like many kinases, it has to be activated. So you have to turn on the signaling pathway by the kinase phosphorylating itself. So the first substrate that FAC has is itself, and it phosphorylates on this residue tyrosine 397. And the important thing to know is that this is a phosphorylation that happens in trans, which means that 2 of these kinases have to interact with and phosphorylate each other. So this is a concentration-dependent reaction. And so we hypothesized that maybe forming a condensate would increase the concentration and then perhaps be a way to activate this signaling pathway. And so we just tested if phase separation could activate this phosphorylation reaction by titrating FAC across its saturation concentration. So again, FAC, if we purify FAC, it is sufficient to phase separate in a test tube. What we see as we sort of in this condition, it phase separates at concentrations above 45 nanomolar. And what we see when we look at signaling is that there's a really big jump in activity when we cross that saturation concentration. So we think that these condensates are really hot spots for autophosphorylation. We can also physically separate with centrifugation the dense phase and the dilute phase, and we see that all the activity is really in the dense phase. Okay, so that's interesting. And another interesting observation, again, is that the concentration of FAC, as I alluded to, is very low in the cell. It's about 5 nanomolar, so lower than the concentration required to spontaneously phase separate. So we think that normally this activation of phosphorylation through phase separation is likely regulated by the receptor. But we also hypothesize that maybe if you overexpress FAC, that would be sufficient to form condensates spontaneously and decouple the phase separation from the receptor. And we thought this might be important because FAC is often overexpressed in a lot of cancers. So this is a Western blot, which is just quantifying the protein concentration in different cells. And in a healthy cell, again, the FAC concentration is really low. You can barely see it in the Western blot. But in different patient-derived cancer cell lines, we see FAC is really elevated in this cell. And so its concentration is increasing by a lot. And it wouldn't take that much overexpression to go from 5 nanomolar to 45 nanomolar. And so we hypothesize that if we just overexpress FAC, maybe we can start to form condensates in the cytoplasm and decouple the signaling from the receptor, which would potentially be bad because you want to regulate signaling in a cell. And so to test this, we just decided to overexpress FAC and see if that was sufficient to induce signaling. We used our knowledge of how FAC phase separates. So this is also, I guess, a hybrid protein that is both folded and disordered. And it's actually the folded domains that are driving phase separation. So there's an N-terminal domain that dimerizes, and that dimerization seems to be important. So if you mutate the dimerization interface, you reduce the phase separation. So there's still some condensates, but it essentially increases the saturation concentration. And then we have wild-type FAC in vitro. And then we can enhance the amount of condensates that we see by adding an additional adaptive protein pexillin, which has multiple binding sites for FAC. So it increases the valency of the system. And this is with in vitro reconstitution. So we decided to use these same mutants in a cell and see if just overexpressing FAC could form condensates. So here we're removing the ligand for the receptor that's supposed to activate FAC. We're overexpressing cells, and we do see in the cytoplasm with wild-type FAC these condensates forming. If we use the mutant that has a higher saturation concentration, we don't see condensates at that same concentration. And if we co-overexpress FAC and pexillin, we see lots of condensates, very similar to our in vitro reconstitution. And so we can sort of also quantify the number of puncta sort of increases as we increase the concentration by just transiently overexpressing to different levels. And then the interesting question was, do these condensates actually activate signaling even though the receptor is not activated? So what we normally see is when you don't have receptor activation, there's a decrease in signaling measured by Western blot analysis of this autophosphorylation site. If we overexpress FAC to levels where we see condensates, we can rescue that even though the receptor is not active. But if we overexpress the mutant that doesn't form condensates, it doesn't rescue the signaling. And we can also overexpress to low levels where we don't see condensates, and that also doesn't rescue the signaling. So there seems to be a correlation where when we see condensates, those condensates are able to induce this signaling pathway even though the receptor is not activated. And so to conclude, we are sort of building our understanding experimentally of how membranes can regulate condensates. What I haven't explored, but I think is also interesting, is condensates are also influencing the membrane. So you can see the condensates kind of changing the local lipid composition. And we saw yesterday how the condensate might also have other mechanisms of sort of changing membranes as well. Membranes contain both proteins and lipids, and both of these molecules can interact with cytosolic proteins that have some propensity to phase separate. So that gives the membrane this really great role of being able to regulate when and where condensates form. in our hands, membrane-associated condensates can activate signaling pathways, at least in the case of integrin. And decoupling phase separation from membranes may lead to dysregulated signaling in cancer. So both, you could imagine, not just cancer, but other diseases overexpressing a protein or mutating a protein to shift its phase separation propensity could sort of decouple this membrane-regulated process. And a lot of the proteins that are sort of seem to be regulated by the membrane are often expressed at very sort of low nanomolar levels in the cell. So I think this is kind of important that the concentration of these proteins is titrated to the correct level so that this behavior can be regulated. And so with that, I'll conclude, and I would love to take questions. That was a little long. Thank you.
Very nice talk. I wonder if the TALEN phase separation can also affect actin polymerization activities?
Yeah, so we're looking at actin now. We haven't looked at polymerization, but we see that TALEN can form condensates with vinculin and actin. So these are proteins known to be associated with TALEN, and we're really interested in actually the viscoelastic material properties. So what we've observed so far is that when you have a condensate that doesn't contain TALEN, so like a FAC condensate is very viscous, purely viscous, but when you start to add vinculin, TALEN, and actin, it introduces an elastic component. And one of the functions of these proteins is to transmit force, and so we're very interested in how all of that is related. Yeah.
We talked, Lindsay. Question on the lipids.
Yeah.
You were saying that kinases can also phosphorylate the lipids, and there's transit potentially between the ER and the plasma membrane. Can you speculate on whether these droplets might be channeling transport fluxes of lipids?
I don't know. So we're really interested if the enzymes that regulate the lipids localize in the condensates, and that could be a mechanism to modify, but we haven't looked at if there's like lipids themselves in the condensates, because that would be wild. And there's also some evidence of like condensates like connecting membrane, like you could potentially connect two membranes together with a condensate, and then maybe that would, So who knows? But yeah, it could be, and it's like potentially more hydrophobic than the cytoplasm, possibly. So there could be weird things going on, but we haven't looked, and we haven't looked at, soluble. All our lipids are in bilayers, so how hard is it to to pull a lipid out of a bilayer is somewhat unfavorable, but maybe possible. Yeah.
Do you think there's any heterogeneity between the 2D part of the condensate compared to the 3D part of the condensate? And
we have not looked at how three-dimensional is it. And so I guess another interesting thing about integrin condensates in cells is they are anisotropic, so they actually have a layered organization that we can visualize in cells, and so that's very interesting. So we don't know, how do you go from a droplet that is isotropic to this layered structure that's quite different from like a liquid droplet. So I think there probably can be heterogeneity. TALEN is also, I guess, another interesting thing is that the lipid binding domain of TALEN is in the N-terminus, and TALEN is 270 kilodaltons long and fully structured, so it's a really long protein. So you can imagine like that itself could introduce some sort of Z difference between the bilayer and the cytoplasm.
That can be used in signaling too.
Yeah, exactly. So.
Thanks. You mentioned topologies at the very beginning, and all of, correct me if I'm wrong, but all of these in vitro is just a flat 2D surface.
Yeah.
And so in your cells, how big are the condensates relative to the plasma membrane? Is it effectively a flat surface? And I guess, is that relevant for the plasma membrane versus obviously intracellular?
So at least where these integrin clusters are forming, the membrane is flat because the proteins are like holding it down essentially. But then like as soon as you go away from that, it starts to become more curved. So yeah, I think the bilayers are both flat and they're sitting on top of glass, so they also can't really deform them. So those are two things that are not physiological. But I'm very interested in curvature and how that might change things. Yeah. Exactly. So yeah, the condensates might be trying to deform our membranes, but they can't because they're sitting on top of glass. Yeah. So.
One quick technical question. In all the images of the membrane, you drew it as attached to a cover slip. are these membranes fluid or do you have any trouble getting them to maintain fluidity over?
Yeah, they have to be fluid, and it's challenging to keep them fluid. But yeah, so yeah, we might think about like so there are other like technologies to look at membranes like GUVs or which have other pros and cons. So our bilayers are definitely fluid. We check that with like photo bleaching. If they're not fluid, so if the glass is at all imperfect, you get an immobile bilayer. And if you have an immobile bilayer, you don't form condensates. So you really actually need the diffusion to.
But you haven't, have you tried to play around with the fluidity in a, like,
not in a
controlled way?
Intentional way, no. But I think that's an interesting question. And in cells, there's like cholesterol and other things that regulate the fluidity of the bilayer, yeah. Oh, yes. Awesome.
Okay, our